Welcome to the 464th Annual Chemathon. We are live here at the CSUN Membrane Stadium. For today's relay race, we have the G proteins, the tyrosine kinases, and the gated ions. Today, our competitors are competing for the fastest elicited response title. Everyone is getting into position. From what I just found out, it is the gated ions' first time joining this race. The G proteins are the defending champions. Let's see if they can win again this year. It looks like our racers are ready to go. There they go. The racers are on their way. Here we have Lenny the ligand running towards Gary the G protein coupled receptor. The ligand hops on the receptor and there is a conformation change. Waiting for the amazing trio triplets, the trimeric GTP binding protein. Oh look, here they come. We see the alpha, the beta, and the gamma sisters running towards Gary, the G protein receptor. We see the alpha and the gamma keep their balance by their lipid tails, anchoring it to the plasma membrane. The trio sisters join Gary and Lenny, ready to pass the signal to the cell. Looks like the alpha sister has to do a conformation change. Luckily, in the plasma membrane, there are plenty of GTPs. GDP comes out. And then one of the GTPs comes in. GTP comes in and replaces it. There seems to be another conformational change. We see the alpha sister break off from the beta and the gamma sister. We see the alpha sister running towards Timmy, the target protein. The alpha sister with the GTP bind to the target protein. The target protein can now head towards the finish line to send the signal to the cell. Meanwhile, GTP hydrolyzes back to GDP, and the alpha sister is now deactivated.
So, here comes our signal molecules, and they will hop onto the binding site of the tyrosine kinase receptor, causing a conformational change. Tyrosine kinase regions are now activated at this point and ready to transfer phosphate groups from Mr. ATP. Our phosphates will now attach to our tyrosines. This molecule is now phosphorylated, a fully activated tyrosine kinase. And now, inactive relay protein number 1 comes up to the phosphorylated tyrosine kinase. When relay protein number 1 is bound to this molecule, it causes a conformational change in the relay protein. This change will lead to a series of events or a signal transduction pathway that will lead to a cellular response. And here comes another relay protein, number 2. It comes up to the phosphorylated kinase. Triggering another cellular response.
The ligand epinephrine is rushing towards the ion channel. The ligand seems unable to bind, so the channel cannot yet move forward. We're getting word that the wrong ligand was sent. What an unfortunate event. I wonder if the correct ligand will make it on time. Oh look, here comes acetylcholine rushing towards the ion channel. And it's a perfect fit. The gated ions are back in the race. Now that the ligand is bound, the ions are free to proceed to continue the race. The ions are heading towards the finish line. It's going to be a tight race. And here comes our racers. It's going to be a close one. Ooh, here they go. Who's the winner? the Gated Ions, the rookies in this race. Congratulations, you are this year's winner for our 464th Chem Annual Thon. Congratulations.